Great, thanks so much. Very kind introduction. I'm super happy to be here. It's not the first time I present in a Good Seminar series. So today we will all be experimenting a bit. I will not do our regular presentation where we present our own stuff. Rather, I'm trying to present a brief overview of what I think might be interesting in artificial intelligence applied to drug discovery. So I've built this presentation from scratch. If something is not perfect, please bear with me. Just a brief introduction. I think we are all between family. We all know each other. I've been at H3D a couple of times already. I represent Ersilia today. We are a tech non-profit that equips laboratories in low-income countries with artificial intelligence tools for infectious disease research. We built our organization based on 3 pillars. The first one is, as you know, like everything we do is free and open source. We prioritize public code, open access licenses, and we don't pursue patents for our work. Most of what we do, at least we try, is in-country research. This means that we try to engage in collaborations that prioritize science led by local institutes, for instance, H3D. And we try to do as much as we can our implementation in situ. This means that we travel as much as we can to the institutes and we implement our tools together. Finally, the third pillar is sustainable collaborations. As you know, or as you might have heard, we are engaged in several capacity building activities. We try to train local scientists, and we try to adapt our tools, our artificial intelligence and machine learning tools, to low resource settings. This is just a nice picture of a workshop that we did last September, I believe, together with the H3D Foundation. As you may know, that was a 1-week workshop titled Bringing Data Science and AI to Infectious Disease Research. Brought together scientists from 7 African countries and was overall like a big success. So hopefully we will continue with this kind of activities and engagement together with the H3D Foundation. So to the topic, what's new in AI for drug discovery in general, and in particular for antimicrobial drug discovery, in my opinion. This is a highly opinionated list. And what to expect from 2023. So I've tried to summarize my thoughts in four broad topics. The first one is large language models. We'll talk a little bit about it. The second one is ultra-large library screening. The third one is a favorite topic of mine, which is targeted protein degradation. And finally, I will talk necessarily about model deployment and data sharing, because I believe that this year we will see a lot of AI implementations that will actually consider this at their core. So let's start with the first topic, very exciting one, large language models. I'm sure you have all heard about ChatGPT. It's all over the news now. The idea of ChatGPT and any other large language model for that matter is that we have a prompt. Let's call it an instruction. This is what the user writes, the human writes. And then there's a generation step where some text is generated by an artificial intelligence. Right? This is not new, but what's new is that those models like ChatGPT can do this exercise with outstanding performance. I don't know if you've had the chance to try GPT, but I really encourage you to do so. For instance, let's try it here. I've written to ChatGPT the following prompt. Tell me how AI can aid drug discovery, because I was lazy, for infectious disease, only 4 items. And this is what ChatGPT answered to me. High throughput screening, virtual drug design, predictive models for disease progression, and genomics analysis. I don't fully agree with this list, but it's actually quite interesting and informative. I think we can all agree. We can do more things with ChatGPT. For instance, I can ask the following thing to this tool. Can you write the above as a poem? And indeed, it's going to do so. So for instance, this is the poem that ChatGPT has returned on AI applied to drug discovery. I'm just going to read, for instance, the first chunk. AI and drugs, a perfect pair, fighting infectious diseases with care. High throughput screening, it leads the way. In the race to cure, it clears the way. So it's actually quite competent as a language generator. And of course, the question is, how can we apply this tool into something that is not only fun, but also interesting for our research. So let's summarize a little bit what's happening in these large language models. And I didn't find any easier way to represent this than this very simple scheme. So as I said, we have an input, the human input. We have a big model, big language model that produces two things. One is the text output, which is what we all see in our prompt. And the second thing is a numerical representation of the text. Both things are equally important when we do research. What users usually care about is the text output because this is human readable. But what data scientists care about most of the times is this numerical representation of the text. What is this numerical representation? It's just a large array of numbers that kind of captures numerically the meaning, the semantics of the text that is being processed. Okay, so whenever we do AI for drug discovery, we are always considering both things, the output itself, but also these latent representations of our inputs. So how does this apply to drug discovery or to biomedicine in general? Well, we can think, for instance, of a particular type of biological entity, which are proteins. We all know about proteins, that actually we can express them as language. How is actually quite straightforward. Proteins are a sequence of characters, and therefore we can think of proteins as language, right? As a sentence. So if we do the following exercise, we can basically adopt all of these great tools that people have been developing in the field of natural language processing to actually understanding and processing protein sequences. And this is what people have been doing extremely successfully over the last couple of years. You can see here again a very simple scheme. We can have a protein sequence. We have huge protein sequence databases. As you know, we have millions and millions of protein sequences out there in the public domain. We can take them as text, which is the input. And basically what we do to train a machine learning model is we hide part of the protein. And we ask as you can see here, and we ask our model to complete the missing parts. In doing this over and over and over millions and millions and millions of proteins, we can learn what we call a latent representation of the proteins that we can use for many, many things. One of them is predicting protein structure. So this is basically the reason why we are having such an explosion of protein structure prediction, AI-based protein structure prediction tools today, one of them being AlphaFold. So let's see how this language model perform in actually predicting protein structures. Those language models are actually extremely fast, so we can just apply them to as many proteins as we want. And here you can see a video where many protein structures have been predicted across millions and millions and millions of proteins sequences available out there. This is what we call the structural proteomes, or the universe of the structural universe of proteins. It's extremely interesting to navigate these predicted universes because we discover known folds as well as new protein folds emerged by these tools. You all know about AlphaFold. AlphaFold is one of such examples. We have many more today. The idea is always the same. We depart from language, that is a protein sequence, and we get as output a protein structure. And as you know, AlphaFold, with all of its limitations, is able to predict some of the structures with outstanding accuracy. As you can see here, in green the known X-ray structure of a few proteins, and then in blue the AlphaFold predicted protein structure of the same proteins, only departing from protein sequence. All right, so why is it useful? Why should we care about all of this if we are doing antimicrobial drug discovery? Well, today people have been actually predicting protein structures for many proteins and putting them out in the public domain. So now we call them structural proteomes of basically any model organism that you can think of, including, of course, human, but also mice, Saccharomyces, etc. And perhaps more importantly, we have resolved structural proteomes of essentially every pathogen that we can think of in the domain of global health, including, of course, Mycobacterium tuberculosis, Pseudomonas, Plasmodium, Falciparum, etc. Right? So it's really, really interesting to browse this database and see whether we can find structures for our proteins of interest. Right. So we've talked a little bit about language models and what we call sequence-based description or representations of the proteins. Of course, we can do much more using numerical tools and algebra in machine learning. We can try to capture also the structural properties of the proteins, and we can even try to capture the physicochemical properties that we can identify on those proteins. The idea is always the same. We have an algorithm that takes as input many instances of proteins and tries to capture their properties numerically, either the sequence proteins, but also the structural properties. Can we do or can we apply the same principle to small molecules? Indeed, we can. And this is actually what people have been doing for a long time with what we used to call like classical descriptors, right? So fingerprints, physicochemical descriptors, etc. Today, people are trying to represent or leveraging text representation of small molecules like the SMILES representation, as you know it, consider it as text or as sentences and try to capture the same way we try to capture the properties of proteins, try to capture the properties of small molecules as text. This is partially successful, and I'm confident that this is going to get only better in the next couple of years. Right, so another tool, AI tool that has been on the news in the last couple of months, or in the last half a year, I would say, is DALL-E and all the derivatives of DALL-E. As you might know, DALL-E is a text-to-image generation tool. So the idea of DALL-E is that you input something like teddy bears doing chemistry experiments, someone at HCD, and then you get an output, an image like that. It's quite impressive, right? So just by asking what you want, DALL-E is going to be able to produce an image that kind of fulfills what you had in mind. Quite impressive. Of course, this is just for the fun. What we really want is something like a novel, cheap, and safe antimalarial compound. Let's see if DALL-E is able to produce something that is interesting. Certainly not, right? So this tool has been not trained and fine-tuned for the domain of interest that we have, which is actually producing nice and beautiful and understandable and plausible chemical structures. DALL-E, this is not the domain of applicability of DALL-E. But it's going to come. I'm pretty sure it's going to come. So there are already some tools. This one is from Microsoft, published last year, where you can actually write a description of a molecule. For instance, my molecule is an 18-membered homodetic cyclic peptide, which is isolated from this particular species and exhibits antimalarial activity, blah, 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 blah. And then the algorithm, DALL-E-like algorithm, is going to come up with a structure that kind of fulfills your description. We are still very far from actually having something that is really interesting in practice, but I think that this highlights that we are all working in the AI field in this direction, right? Having an input from a researcher that is going to be able to produce an actionable and tangible output like a molecule structure. This is actually perhaps more developed and in the field of proteins rather than small molecules, for the reason that proteins can more naturally be expressed as language. So I'm here just highlighting a paper published recently in Nature Biotechnology, where they actually do an impressive job in trying to leverage this concept of natural language processing for designing new proteins. So the concept is very simple. The concept is working with this idea of tags, right? So we can have different tags that can kind of narrow down the domain of applicability of a particular prediction, right? And imagine we have this initial text like the dissolution of. So if the tag is chemistry, a likely output is going to be salt and sugar. But if the tag is politics, a likely output can be Yugoslavia or Parliament, right? So by using these tags, we can kind of orient our machine learning model towards the type of predictions that we are really looking or expecting, right? If we apply this to protein sequences, we can think of like, let's say we want to design a protein. We can start with a sequence like that, typical sequence for starting a protein. And then we can have the different control tags, for instance, the function of the protein. And by doing so, we will hopefully be able to design with this molecular protein that will fulfill this particular sequence. They have done this with enormous success in this paper, and they've done like quite massive validation. I really encourage you to read this paper. The idea is that they provide a tool where you can actually ask for a function, and the molecule is going to be able to hopefully predict proteins that will, enzymes, for instance, that will be able to fulfill that function. And here they are showing this in, for instance, for this, let's say, antibacterial peptides that can be useful, right? Pesticins, phage lysozymes, glucosaminases, etc. In red, you can see natural proteins, so proteins that really exist, and in blue, you can see artificial proteins, so proteins that the machine has invented. As you can see, they fall within more or less the same sequence space, which means that they are actually quite likely to exert the function that we expect, and indeed they did quite massive validation in this paper. Can we do something similar with chemistry, or have we even tried? Well, we have tried, even if we are not quite advanced as in the previous example. Here you can see, for instance, a prediction of different chemical structures that us at Ersilia tried to do for antimicrobial activity against the Plasmodium falciparum parasite. You can see a common core that we know is active, and here you can see in the branches different, let's say, derivatives of this core that we are trying to suggest that we believe can be active, right? The more blue you see here in the background, the more active we predict those proteins and those small molecules to be. At the end of the day, this is something that we can actually do with these generative tools, language-based generative tools. At the end of the day, we can produce a large set of candidates. In this case, we have 100,000 candidates that we produced over an afternoon in a laptop computer that we can explore and basically pick from there like small molecules that we believe might be interesting for this particular assay. In this case, this was done in the context of a consortium that I'm sure you all know, which is called Open Source Malaria, led by Matthew Todd. We suggested, I believe it was 8 candidates from this large pool of candidates that we produced with our machine learning tool. We selected 8 of them. They tested eventually all of them. And out of the 8, 4 had submicromolar activity, which was quite satisfactory. The same thing we can do with other exercises with, let's say, the similar amount of effort. In this case, in the context of open source antibiotics, of the Open Source Antibiotics Consortium, we tried to do something slightly different. In that case, we were not optimizing for, let's say, a whole cell activity, but we were optimizing for a accommodation in a particular binding site that was desired in a particular enzyme of Staphylococcus aureus. So in that case, our score for optimizing those molecules was a docking score. When we did that generation again of different structures, we came up with about 700 compounds. Almost 200 of them were purchasable already from Enamine, and we actually have purchased, I think, 25 of them, and they are currently testing them at the Open Source Antibiotics Consortium. So we are getting there slowly. Which brings me, I think, I hope quite naturally to the second topic that I would like to highlight today for 2023. I think that this is something that we have all heard about, the concept of ultra-large libraries, and how can we leverage AI for effectively screening those libraries. So as you know, we all know about this, the chemical universe is huge. When we think about like public data, which is mostly what we deal with at Ersilia, the number of molecules that we have is relatively limited. Let's take PubChem as an upper bound, 10 to the 8. Then if we go to zinc, We have already 10 to the 9 molecules available out there. And if we think about commercial libraries like enemy and real, we can go up to already tens of billions, right? 10 to the 10. But of course, if we think about companies like, for instance, GSK, I think they have like the largest proprietary library available out there. Is 10 to 26, so extremely huge libraries. So the question is, how can we possibly screen those libraries in a way that is efficient and reliable as well? Well, AI proposes like some solutions to that, especially applied to this concept of make-on-demand libraries. So make-on-demand libraries, I think that the most common or well-known example is the enemy real library. Those are libraries that are built basically based on some building blocks and known reactions. And the idea is that they do not have those molecules physically. They are not purchasable, but they know how to synthesize them in a short amount of time, let's say under 3 weeks. So basically, NMIN enumerates all the possible compounds that they can possibly synthesize, and it provides them publicly, so we can all screen those compounds, come up with some hits, and go back to NMIN and purchase those compounds and eventually test them, right? So this concept of make-on-demand libraries is actually becoming quite popular, and many of the AI tools that we are developing are leveraging those libraries to actually speed up the screening. How big are those libraries? As I said, over 10 billion compounds, so actually quite big. Between 2021 and 2022, there was like a stream of papers in Nature suggesting ways of using docking in combination with AI in different flavors to actually screen those libraries efficiently. Yeah, the idea is that, of course, we cannot do docking. Realistically speaking, many labs cannot do docking with so many compounds. So we want to use an AI tool that kind of aids us in making this screening more efficient. And like, to cut a very long story short, usually what we do is we couple, like, the docking experiment is what's considered to be the most reliable thing, but we couple the docking experiment with a pre-filtering step that is AI driven. And this has been shown to be quite successful in a couple of instances. Most of them apply to the enemy in real library, as I was saying. Okay, let's switch gears a little bit. I want to talk about a favorite topic of mine, which is targeted protein degradation. I think that the most famous family and successful so far family of compounds within this topic is our products. I'm sure you have all heard about it. Let me just summarize a little bit what's this concept about. The concept of targeted protein degradation, we can call it a new paradigm for therapy, which is basically the idea of actually not inhibiting.
More and more interesting. One of the papers will be talking about T-cell cloning.
Sorry, please
Is this a question?
mute. What
Okay.
Perfect. Thank you.
No worries. So the concept of targeted protein degradation, it's actually a new paradigm in, let's say, in medicinal chemistry, where the idea is not to inhibit the activity of a particular protein, which is typically what we want to do in, let's call it, occupancy-driven pharmacology. Here, the idea is to actually trigger the degradation of that protein, right? And this is usually achieved by bringing together we call it protein of interest, with the degradation machinery that we natively have in human cells, right? And this typically involves basically favoring the interaction between an E3 ligase and our protein of interest with a protax that is a small molecule that is able to stick those two proteins together. And the way this works is with like a mechanism like this. This is our protax. It brings together here in green the protein of interest, the degradation machinery. It brings together the E3 ligase here with our protein of interest, our protein gets ubiquitinated and then it's recognized for degradation, right? This is a very interesting new therapeutic approach. And even if we don't have a lot of clinical evidence yet for its success, it's certainly very interesting in terms of expanding the druggable genome. So the question is, okay, we have a new paradigm. How can AI help? So we recently published a paper, a perspective in JACS, kind of reflecting on how AI can contribute to targeted protein degradation. The idea, as I was saying, is targeted protein degradation addresses some of the, let's say, old challenges of occupancy-driven pharmacology. That is, some proteins do not have a, let's say, a binding site naturally, or an orthosteric site, or even an allosteric site. As we know, occupancy-driven pharmacology has selectivity issues, and sometimes our binding is non-functional, right? So all of these challenges of, let's say, developing inhibitors that we all know are kind of addressed with this new paradigm of targeting protein degradation. However, this opens new challenges. For instance, how do we discover ligands that are amenable for targeted protein degradation? Which phenotypic assays should be doing? And how can predict structurally whether a certain product is going to be able to bring together the protein of interest and the E3 ligase as we want, okay? So all of this, of course, the field is quite new and we don't have a lot of tradition, a lot of data science, a lot of computer-aided drug discovery tradition in these fields, and we need to ask how can we help with AI? So I anticipate that a lot of the AI work that is going to happen in drug discovery this year is going to revolve around this concept of targeted protein degradation. Just to focus on one example, you can again refer to the review for the others. I think it's quite interesting to consider how we model. We call them ternary complexes, right? The ternary complexes is the interaction between the protein of interest, the E3 ligase, and our small molecule. Here you can see products which are, let's say, first generation of targeted protein degradation molecules, but we can also do exactly the same thing with molecular glues. Molecular glues are smaller, are not bifunctional. Products usually recognize the E3 on one side and the protein of interest on the other side. Molecular glues are not bifunctional. They do the same. They are more sticky, and they do the same thing actually with the core structure. In any case, designing those molecules with AI is going to be challenging because we do not have a lot of training data, because we do not have a lot of instances. So the design, we anticipate that the design of products and molecular glues is going to come at the interface of structural biology. So we will have to be able to apply the structural tools, hopefully the AlphaFold predictions, together with ultra large scale virtual screening, and to actually be able to discover small molecules that can successfully become products or molecular glues. Why is it interesting? Why is this concept interesting also for antimicrobial drug discovery? Well, last year, a very similar concept of products was proposed for bacterial systems. That was called BAC products, published in Cell, and basically what they suggest is so the degradation machinery as we know it is obviously not the same in bacteria than in human. So these people have proposed a different system that can more or less analogously be applied for targeted protein degradation in bacterial systems. And I think that in this paper, they showcase it for mycobacteria, which is quite interesting, of course. Okay, this brings me to the last, I believe, interesting topic that is going to come, and we are going to see a lot in 2023 with AI applied to drug discovery, which is model deployment and data sharing. Why I think it's interesting? Because we've had, quite honestly, an avalanche of startups, companies, even big pharmaceutical companies opening AI departments. And there has been, for the last couple of years, there has been a lot of interesting research and a lot of developments, and now it is the time to make all of those tools available to medicinal chemists, biologists, etc. So there's a component of AI which is usually unperceived, which is how do we make those models available? How do we turn them into actually user-friendly tools that we can share and we can use in our daily lives? And this is actually at the core of our organization. Actually, what we do at Ersilia is we try to bring all of those amazing tools that people are developing into something that is usable for all of you, so that you can incorporate them in your daily research. So our ecosystem at Ersilia has three main components. One, on the one side here in the left, you can see the Ersilia Model Hub. That's our main asset. The Ersilia Model Hub is a repository of pre-trained, ready-to-use machine learning models that researchers can use, hopefully easily in their daily tasks. Here in the middle, you can see something called Zyracam, which is an automated machine learning tool that we have developed, and I'm proud to say that we have developed this in the context mostly of H3D, as you will see now. And finally, here on the right, this is something that is still work in progress, but we have to worry about privacy, we have to worry about data sharing, etc. Because if we really want to actually bring all the data that we are producing and all the assets that we are producing together, especially at the intersection of private and public enterprises, we need to be sure that all the legal issues that might be associated with it are preserved. And this belongs to a field that is called privacy preserving AI that we are also exploring quite intensively in the context of a Merck-funded project. So back to the Ersilia Model Hub. So what do we mean by deployment? The idea is very simple. The idea is that when we do AI, there's a lot of things that are going on inside the algorithm, let's say. Historically speaking, let's say we had some raw data that we had to process, we trained the model, we tested, validated it, we did some external validation, etc. So all of this is not necessarily interesting to the user, right? What the user wants to see is what we call the deployment layer, which is just a tool, an interface where they can input something, for instance, a drug, and get something as output, for instance, the predicted targets, right? So this outer layer is usually not very well developed in academic research because publication stops before. So our mission as an organization is to provide this outer layer to as many models, published models as possible so that they become useful to everyone. This is a snapshot of the Ersilia Model Hub. We are aiming at over 100 models in the next few weeks. We are already like at 95, I believe, at the moment. You can see here that we have models of different categories related to toxicity, antibiotic activity, synthetic accessibility, antiviral activity, etc., etc. You can browse it, and then if you click in one of them, you will find instructions on how to run them. How do we achieve all of this in a way that is scalable? As you know, we are a very, very small organization. So how do we achieve this in a way that is actually sustainable and scalable, and most importantly, can be up to date with the literature? We have come up with a open source community workflow that we really rely on. This is the idea where spontaneous contributors can emerge from all over the globe, and they can do their contributions. And for ensuring that, you need to have an actual workflow that ensures that the quality of the models that we are incorporating is good enough, and that everything works and is tested. So how this workflow works is we have a user that has a new model idea. Let's say that they have found a paper in Cell that provides a machine learning model. They raise an issue in our public platform. There is discussion about this model. At some point, our core team approves the incorporation of this model. And when this happens, basically there is a coding infrastructure that is opened for this user where they can incorporate the model in their own computers. And at some point they will say, okay, I'm ready. I'm going to give it to you now. And then this model is submitted, we approve it. There's some automated workflows that happen in our cloud. Then we do some clerical review, leveraging our community, and when everything looks good, we release it. Only when this whole pipeline is completed, the model is eventually published in our resource. So we can call our resource well curated. Let me show you an example that hopefully you will care about, which is the incorporation of some of our models in the virtual screening cascade as a virtual screening cascade at H3D. So, I mean, I don't have to introduce H3D here, but basically, I think that what's important to note here is that we have leveraged the data that has been accumulated in your institute in your center for over 10 years. This is an enormous and super valuable amount of data that we have tried to convert into AI models that you can use prospectively for making predictions for new compounds. The idea here is that we build models together with our colleagues at H3D. Those models are deployed, and therefore, when new molecules are designed, they can be predicted with the models before doing the actual experimental test. So at the end of the pipeline, there is a very, very complex, let's say, dashboard that would look like that. Here I'm just showing the example of Mycobacterium tuberculosis assay, and we have, like, we call it a results canvas. What's important, I believe, here is to focus on this particular plot, where you can see that the model output is a score here in the y-axis, and in red you see known active molecules that were never shown to the model before, and in blue you can see known inactive molecules. And what's interesting to note is that in general, known active molecules receive higher scores than known inactive molecules. This means, in practice, that by using these models, we are able to successfully prioritize some of the compounds, reducing the attrition rate. For how many assays have we done this kind of exercise, and can we produce this kind of canvases? It's for about 15 assays, departing from the 13,000 molecules that we could collect over the last couple of years at H3D. And those assays belong mostly and primarily to wholesale assays from Plasmodium falciparum, Mycobacterium tuberculosis, and we are doing some explorations in AMR, as you might know. There's also cytotoxicity assays, and there's also some of the ADMET assays. Overall, not all models work equally well, of course, but we are quite satisfied with the results. Here you can see the panel of assays that we are now that are now deployed at H3D. This includes, as I said, like Plasmodium, Mycobacterium, and some of the ADMET assays. And you can see the kind of plot that I was highlighting before, where actives and inactives are nicely stratified by a score produced by the model. Let me just show you here a prospective example where we actually predict some compounds that belong to a particular series experimented here at H3D, and we produce for each of these compounds, in this case 65 compounds, different scores related to desired, let's say, properties, that is usually antimicrobial activity, and undesired properties, for instance, interaction with HER. And by looking at these profiles, predicted profiles of desired and undesired proteins, desired and undesired properties, we can kind of hopefully pick the compounds within a series that we believe are more interesting. All right, so I think that this is all I wanted to say about deployment and data sharing. Just a little bit more about our organization before we wrap up. Ersilia is a non-profit organization aimed at developing machine learning tools for drug discovery and global health. As you know, we combine remote work and on-site implementation. We leverage capacity building activities, and pretty much everything we do is open source. I'm extremely thankful to many of the people here at H3D and also to our donors and collaborators. We've been very lucky to grow quite fast in the last couple of years, and this has only been thanks to all the collaborators and supporters that we've had. And I think that this is all right on time, so I'm open for questions and discussion. Thank you very much.
Perfect. Thank you very much for that wonderful talk. I see we already got some questions going, so you definitely incited some interesting discussions. So I think let's get straight into it. So first, I see we have a hand from Kelly, or are we clapping? Not too sure.
Sorry, I meant to clap. My apologies.
Okay.
I meant to clap. Sorry, I misplaced. Sorry, Miguel. Yeah.
No problem. Okay, perfect. Okay, we have a few hands going up and down. I'm not so sure. I think that might be some people trying to clap, but please, if you do have any questions, please raise your hand. Or you can speak now because I'm not too sure. There seems to be a bit of an issue.
There
Okay,
are some questions in the chat, Sonia.
Is there? Perfect. Thank you. Okay, we have one question from Vin. So he asks, considering most of the AI tools / outcomes are built on open source data, please provide some light on developing or implementing IP rights for the drug AI development.
Great. So thanks, Vin, for this is a very important question. So what I can say is what we have been doing, which falls within this field of, we call it privacy preserving AI, as I was saying. So the idea here is that we have to ensure privacy for two actors. One is the data provider, right? So people might be willing to contribute their data to a machine learning tool, but they don't want their data to be revealed eventually, right? Of course, understandably. This is the case for many of the pharmaceutical companies. So the guarantees that the encryption guarantees that we need to give in this case is that if I provide publicly a machine learning tool that has been trained on private data, I need to guarantee that a malicious user will not be able to reverse engineer your molecules, right? Because if this happens, then your privacy is violated. So this is achieved with a type of encryption that is called homomorphic encryption, and we are developing, thanks to the support of Merck, we are developing a library for ensuring that. That's only one actor, though. There's a second actor that usually wants privacy and that we have realized and we were not expecting, which is the user. Imagine I deploy a machine learning tool, for instance, for antimicrobial prediction, and you are working with some molecules that are private. If I'm deploying that as a web service, most likely you will not be allowed to submit those molecules to the web service, right? So this we need also to guarantee end-to-end encryption for that, for these type of users, which means that not even us who are offering the web server are able to see your input. Both things can be guaranteed. This comes at the expense of the models being slower, but this can actually be done. And this is something that we have been developing. We don't see how much adoption it's going to have, but we do believe it's an interesting direction, especially to ensure participation of private partners here, right?
Perfect. Thank you very much. I do have one question. I wanted to know, do your models, because you're predicting a lot of compounds, they're not always that easy to make synthetically. So do your models incorporate synthesizability in any aspect, or?
Yes.
Yeah.
So we have, I believe it's 5 models for synthetic accessibility, retrosynthetic accessibility, and even for synthesis planning. This doesn't always work, but at least gives an idea of how accessible synthetically a compound can be. And this is certainly one of the scores that we always consider, synthetic accessibility of compounds.
No, thank you very much. Yes. Are there any further questions? Okay, it seems like we don't have any more questions. So I think you can reach out to Miguel directly. He did have his details up, or you can contact us and we can share those details with you. But that was a wonderful presentation, Miguel. Thank you very much. You never cease to amaze us. And we look forward to hearing from you again soon.
Thanks so much. Bye, everyone.